All right, so here we go with my step-by-step walk you through how to set up a genetic problem. These are what we're going to call these Mendelian genetic problems, Mendelian inheritance problems, something like that. Now, this setup is actually my husband's, and so I'm not 100% sure how this is going to go, but just bear with me, and I think it'll get better as I get more practice with it. All right, so now in the beginning here, we're just going to talk about just what I call straight up or classic Mendelian experiments, okay? Where we're talking about the things that he did with his pea plants that led him to the conclusions that he came up with. Now, in his experiments, he always started with true breeding parents. Now, the term true breeding is just another way to say homozygous, okay? So true breeding meaning that that parent will always breed true to its color, size, height, or anything like that. All right, so here we go. So this is what we have. We've got a true breeding purple pea plant is crossed with a true breeding white pea plant. And what we want to know is information about their offspring. What are the possible genotypic and phenotypic ratios of their offspring? Now, basically, the genotypic ratio is going to be what possible combinations of these alleles could we expect in the offspring? And the phenotypic ratio would be what colors do we expect to see in the offspring? Okay, all right, so now I'm going to go, again, real, stickler to my steps for solving the problem so you can see how we kind of walk our way through them. And in the beginning, if you're not comfortable with doing these types of problems, I would absolutely tell you to follow my steps and write everything down until you get the hang of it, okay? All right, so remember, step one is always read the problem. And read it twice, at least, okay? You're gonna skip over terms, phrases, that type of thing, and it's just gonna be a pain in the butt. But you need to read everything twice. You need to underline perhaps what you're given, okay, so that you know what you're dealing with. All right, now step 2 was the figure everything out step. Now for step 2A, we wanted to figure out the trait that we're dealing with and the alleles that we're dealing with. Now in this case, we're dealing with the flower color and the two alleles are purple and white. Okay, and so purple and white are the two alleles. Okay, then the next step is we want to figure out who's dominant recessive. So who is dominant and who's recessive, and assign letters. All right, now the letters are really just going to be like a symbol, right? For, okay, this is the purple allele and this is the white allele. Now, now in this case, what we know is we know that the dominant allele is purple and the recessive is white. Now, how do I know that? Well, after many, many years of doing all these problems, I basically remember. You also may be given this information in the problem, or there may even be a chart. Okay, I know in the lab that, you know, there's like a chart that, you know, gives you these characteristics, the traits, and who's dominant and who's recessive. It may be that the question reads, a true breeding purple pea plant who is dominant for flower color is crossed with a true breeding white pea plant, comma, the recessive color. You know, so it might be as blatant as you know, just to literally tell you that in the sentence, or it's something that you'll have a chart to refer to, and then after you do enough of these problems, you'll just kind of just remember, okay, in pea plants, the purple is the dominant color. In pea plants, for the trait of height, tall is always the dominant allele. So you'll pick these up as we go. Now, so far as assigning letters, this is my rule. Okay, keep it simple so that you don't get confused, and always go with the first letter of the dominant allele. So in this case, what letter am I going to use to represent purple? It's going to be a capital P. And what letter am I going to use to represent white? I'm going to use a lowercase p. Okay. Now, honestly, you can do whatever you want to do. Okay. But keep it simple and stick. In this case, I'm going to be sticking to one letter per trait. Okay. Now, in this particular problem, we're just dealing with one trait, with just the inheritance of one trait. But we're going to look at problems where you have the inheritance of multiple traits. So you're going to have multiple letters. And when you've got 4 or 6 letters you have to keep straight, it gets a little crazy. So keep it simple. You're going to assign a letter for each of the alleles. Keep it the same letter. And which letter? Just go with the first letter of the dominant allele. Okay. The capital form is dominant, and then the lowercase always goes with the recessive. Okay. All right. Now, step 3 is to write down the genotypes of the parents. All right. Now, one parent is a true breeding purple, and the other parent is a true breeding white. Okay. Now, remember, the genotype is the combination of alleles that this parent is going to have. Now, this purple parent clearly has to have a big P because it's purple. It has to have at least one. But is it big P, or is it big P, little p? Is it big P, or is it big P, little p? Well, either of these genotypes will give you a purple parent. But because you're told that this purple parent is true breeding, which is another way of saying homozygous, we know that purple parent can only be big P. Now, our white parent obviously is going to have a little p, and the only way to be recessive is to be, now I never know quite how to do a lowercase p so that we can, okay. The only way to be recessive is to be homozygous for it. So you're always going to have little p. All right. So what this looks like when we kind of like would write this out in a problem is it would be this. We have a true breeding purple crossed with a true breeding white. Okay. All right. Now let me slide this out, slip a new piece of paper in here. Okay. Now we have step 4, where we're going to figure out what kind of gametes each parent can produce. Okay. Now, remember, to get the gametes, we have to go through meiosis. Okay, and meiosis, remember, is a reduction division. Okay, the whole point of meiosis is to separate the pair of homologous chromosomes into separate gametes so that you go from a diploid parent to a haploid gamete. And meiosis happens in 2 steps. You have meiosis 1 and then you have meiosis 2. So what I'm going to show you here is what I kind of refer to as like this gamete tree, which is kind of it's not exactly a shortcut because it takes a couple seconds. But it's kind of a way for you to visualize this separating and just visualize meiosis. All right, so this is my pair of homologous chromosomes. Now, you know that during the S phase of interphase, our chromosomes duplicate and the original pairs up with its copy. So here's my original pair, and here are their copies. Okay, so here we are entering prophase 1 of meiosis 1. Now, as we go through prophase 1 and the crossing over, and we go through meiosis 1, we come to anaphase 1, and in anaphase 1, we separate the homologous pair. And then we go through telophase 1, and we end up with 2 diploid cells. With 2 haploid daughter cells. Now, and this is where, and just like my slip of tongue there, again, it's like, I see 2 chromosomes. What do you mean this is haploid? Well, remember, we don't count the copies yet. Okay, so this is meiosis 1, the reduction division, where we separate the homologous pair. Then meiosis 2 is about separating our duplicated chromosomes into individual chromosomes. So this is where the sister chromatids separate. So this is meiosis 2. And here are our gametes. Our purple parent can only produce gametes that would contain a big P. Now, this is my white parent, and I would go through the same. So here is entering meiosis 1. Here is the reduction division of meiosis 1. And here is the separation of my sister chromatids, my duplicated pair, into independent individual chromosomes. And so here are the gametes that my white parent can produce. My white parent can only produce gametes with little p's. Okay? Now, hopefully you know this, but sexual reproduction is when you take an egg and it is fertilized by a sperm cell, and that resulting single-celled organism is either called a fertilized egg or a zygote. Z-Y-G-O-T-E, called a zygote. All right. So, so if the egg has a capital P and the sperm has a lowercase p, then the zygote would look like this. So this is your haploid gamete. This is your diploid, chromosome numbers restored zygote. Okay. All right. So now, so this is sexual reproduction. This is the whole point of it, is to combine or recombine the genetic information in the female with the genetic information in the male, and this is your new combination of genetic information. All right, so the final thing then that we need to do for our genetic problems is we need to set up and fill out a Punnett square. All right, now when we're dealing with one trait, with the inheritance of just one trait, that's known as a monohybrid cross. And with a monohybrid cross, you're going to start your Punnett square is going to be a 4-square Punnett square. Now, in this case, I'm going to say, and I'll say this 100 times in the next week or so, make your squares big. Make sure that you have plenty of space in here to write your letters, your capitals, your lower cases, so that you're not trying to cram everything together. Okay. Now, so you're going to draw a square divided into 4, and then we're going to have this diagonal line going off the side. Now, this diagonal line, this is where you write down the parents' genotypes. Now, there's no rhyme or reason, there's no rule what parent goes on top, what parent goes on the bottom. I just kind of traditionally put a dominant parent on top and then a recessive parent on the bottom. But it might be that I'm crossing 2 dominant parents, so whatever, but just put one parent on the top, the other parent on the bottom. Now, what goes right here and right here are the gametes of the parent on the top. And what goes right here and right here, again, are the gametes of this parent on the bottom. All right. Now here's kind of a tricky thing. In a monohybrid cross, there's only a possibility of you getting up to 2 different gametes. Okay. So we only have 2 places for a gamete to actually be written down. Okay. So that's why it's a 2 by 2 Punnett square. Okay. Now, in some crosses, you may only have 1 possible type of gamete. Okay. And this cross is actually, one of those. So some people say, oh, well, I could just take a shortcut and just do this and, call this my Punnett square. You could, but then you might get into the habit of only making it a 1-square Punnett square. And when it comes time to have a 4-square or a 16-square Punnett square, you kind of forget. All right. So keep it simple. Always start off with a 4-square. For monohybrid problems, always draw a 4-square Punnett square. Okay? All right. Now, so now this is where things get a little touchy, because now what we have to do is we have to take the gamete from the purple parent and we have to combine it with a gamete from the white parent, because that's what happens during sexual reproduction. The egg and the sperm combine. So if we pretend that this is the male and this is the female and this is the sperm and this is the egg, we need to bring these 2 together and this is the offspring. All right? This is our new combination of genetic information. All right? So we do that for all the boxes. All right? We combine the gametes. All right? So all 4 of these boxes, these are the offspring. What Mendel called the F1 generation. The first filial generation. The first generation of offspring. Filial basically refers to child. Okay, so these are the parents. This is the P generation. These are their offspring. This is the first generation of offspring. Okay, so you can see how the Punnett square gives you this visual representation of the possible combinations. Now, your genotypic ratio. All right. Now, a ratio is a comparison. When you compare the number of this to the number of that. Okay. So ratios are always written in kind of this particular style, and I'll show you that. So now in this case, for the genotypic ratio, we need to look at the genotypes of the offspring and figure out how many of them that we have. Now in this case, you can probably tell that we only have one type of offspring. They're all heterozygous, big P, little p. So our ratio looks like this. It's 4 heterozygous, and then you do a colon right here, or a semicolon, or a colon. You do the 2 dots, and then it's 4 heterozygous to 0, to nothing else. Okay. Now, sometimes people will just write down that instead of putting it in a ratio form, they'll just say that they're all heterozygous. But if we're sticking to being, to ratios, it's 4 to 0. They're all heterozygous. There's nothing else. Now, if you wanted to write down 4 heterozygous to 0, you certainly could. But it's just simpler and a little quicker just to write down big P, little p, for that. All right, so that's our genotypic ratio. Now the phenotypic ratio, remember the phenotype is the is the physical description of the offspring. So in this case, the question is, what color are the offspring? They are all purple, so we could say 4 purple to 0, or you could say that they're all purple, or you could say that they're all homozygous dominant. Okay, so it gets a little crazy. But just, remember, keep it simple, and I always just put everything in a ratio form because that's just the habit of that. Okay, now where we are right now is we are showing what Mendel saw whenever he would cross, his pea plants and everything. He'd be like, all right, I crossed a purple with a white, and so here's a purple with a white, and I get all purple. I don't get any white, Now, what Mendel didn't know about, and what he certainly couldn't see, was that this little p, this little white allele, it was actually still there. It was just being covered up by the big P, or by that purple allele. All right. So Mendel was wondering, well, what happens to the white color? Like, is it gone forever? Well, no, because there's still white pea plants around. So he wondered, what would happen if I took 2 of these plants? If I took this plant and this plant, and I crossed them, he was wondering, what would happen if I take 2 purples and I cross them? Am I going to get all purple again? All right. So he was trying to figure out what happened to the white. Okay. Now remember, he didn't know about these genes and alleles and stuff like that. He literally was basing everything just on color. But he was curious as to where the white had actually gone. So what Mendel did is he would carry this. He did what's called a self-fertilization, or he would carry the cross. So we're going to say carry this cross through the F2 generation, which basically means you're going to take two F1 offspring and cross them. This is also known as self-fertilization. Now, if you're thinking this is a little odd, you're right. It's like we're taking two siblings and crossing them. But we are doing this with plants, which is perfectly fine. The way plant genetics work, this isn't a problem. You cannot do this with animals or humans. Our DNA does not like to be combined with DNA that's too similar to it. But in plants, and like other organisms, probably bacteria and fungi and stuff like that, this self-fertilization technique is perfectly fine. All right, so this is what we're going to do. So we're going to take two of those F1 offspring. Now it doesn't matter which two because they're all the same. They're all big P, little p. They're all purple. And we're going to cross them. Now, remember our steps. Now, I think we can check off steps 1 and step 2 because we know. We know that big P stands for purple, and we know little p stands for white. Okay, and we know the genotypes of the parents. Okay, good. Now, that was step 3. So BP times PP. All right, step 4, the gametes. The gametes of each parent. All right, now it's no different than how we figured it out for the homozygous parents. Now we just have a heterozygous parent. So what we do is, so this is our diploid, this is our pair, this is our, our homologous chromosome, our pair of homologous chromosome. We're going to duplicate, and the original pairs up with its copy. And then this goes through meiosis 1, where we separate the original chromosomes. Now, but they are still connected to their sister chromatid, right? And so meiosis 2 separates our original chromosome from its copy. So this heterozygous parent is going to give us different gametes. Okay? Our homozygous parent could only give us the same type of gametes. If I slip this in here, okay, this is our homozygous parent. Look at the gametes. They're all the same. Our heterozygous is going to give us different gametes. So now when we set up our Punnett square, now remember, this is a monohybrid cross. It's a 4-square Punnett square. Okay. And the parents, these two, they're now going to come here and here, here and here. All right. And now for their gametes. Now in this case, I have 4 gametes, right? But I'm only going to pick 2 because I can't fit 4 up here, right? So the idea is that you want to represent each possible gamete. So I'm going to take one of the big P gametes, and I'm going to take one of the little p gametes. And then the same thing happens for that parent. Because this parent, if, it would just be this whole, diagram again, okay? And now I combine them. Okay, and now these become my F2 generation. the second generation. All right, the parents were my true breeding purple and my true breeding white. Okay, and then their children were called the F1 generation. And then I took two F1s and I crossed them, and so these are the F2 offspring. Now, check out the genotypes. So here's our genotypic ratio. Now, as you look at your genotypes, and they're right there in front of you, okay, you're like, uh-oh, I have different genotypes. How many different genotypes? Well, here's one, and here's a second one. Now these two are the same, and then here's my third. So I actually have three things in my ratio. I'm comparing three things. Now how I'm going to write it, we write homozygous dominant first, and then we write the heterozygous, and then we write the homozygous recessive. And then you just simply write down how many numbers of each do you have. So I have one homozygous dominant, two heterozygous, one homozygous recessive. Now your phenotypic ratio, remember, now the phenotype is just the physical description of the offspring. So what color is this offspring going to be? Purple. What color is this offspring? Purple. Remember, in Mendel's genetics with his pea plants, dominant was dominant. Whenever you had just one of the dominant alleles, that's what the color was of the offspring. Okay, so these three are going to be purple, and this is going to be white. So my ratio becomes this, 3 purple to 1 white. Okay, now, again, did any of this support the blending theory? Did we ever get a lavender or a pink colored plant? No, we got purple and white, or we got all purple. Okay, now we haven't ever gotten all white, but Mendel, I mean, that's okay because our parents were always purple. Only way you're going to get white is if you cross two white, plants. But this at least kind of proved to Mendel that there was the blending theory was just wrong. Okay, we don't have a blend, of in pea plants. This blending theory was wrong, and this led him to this conclusion of one of his laws that we'll start talking about them in our next lecture. So what I want to leave you with right now is just this technique, if you want to call it, for how to set up these monohybrid problems. Okay, this is not snap your fingers and boom, you're done. Okay, this is not look at this and reason it out in your head type of thing. For the lab assignments that you have to do in order to receive credit, you have to show your work, and this is your work. Okay, at the minimum, this is what I have to see in order to show all of your work. Now, if you, like me, I always have to write everything down because I just can't keep a lot of information straight in my head. So I'm going to encourage you, write everything down, especially in the beginning, until you really get the hang of these problems. Okay? All right. I will see you in our next lecture about patterns of inheritance, where we take it up a step. See you then.